dive in, I'd like to thank today's sponsor, Apis Tactical. Apis Tactical is a new American beekeeping brand that is bringing innovative designs to beekeeping protective gear. They just launched a new glove and there is more to come. Sponsorships like this help me freely talk about important topics in the beekeeping industry. Thanks Apis Tactical for the support. Please check them out, link in the description. Hello everyone, this is your host, Dr. Umberto Boncristiani, and in today's episode, I'm thrilled to introduce a very special guest, Dr. Kirk Anderson, a leading researcher at the USDA Carl Hayden Bee Research Center in Tucson. Dr. Anderson has been at the forefront of understanding bee health, particularly in the context of microbial communities and honeybee pathogens. In this episode, we will explore some critical and timely issues in honeybee health, including his recent work with Randy Oliver on the efficacy of non-native probiotics in honeybee colonies, his strong letter against the use of non-native probiotics in honeybee health, published in Bee Culture magazine, and other topics. And now, I introduce to you Dr. Kirk Anderson. Dr. Kirk Anderson, thank you very much for joining me here today. It's a pleasure to have you in my show. How are you?
Oh, good. Thank you very much, and thank you for having me on.
Oh, it is. I was bombarded by emails and requests to talk to you and to interview you, especially after the publication of you and Randy Oliver published an article about probiotics, and you guys didn't find any evidence on that. I want to discuss this research that you did and the whole probiotic kind of discussions that are circulating out there. You know, is effective? It's not effective. Do you have evidence? We don't have evidence. And you are doing this for so long. I wanna pick up your brain about things that happens in the field. But before I jump into this, I want to know a little bit about you. How you get into the bees? You know, how you end up there at the Tucson and starting and working with bees. Can you give us a little overview about your career?
Sure. The real answer is I have no idea, but I'll tell you the story. So at Boise State, I finally entered school. I was about 28 years old when I decided to go back to school. And we went out for an ecology class. I got really excited about ecology, and we went out to look at this ant species. And we did all these measurements, and we found out that this ant species was over-distributed and it had these colonies. And then I started learning about colonies, and I read Arthur C. Cole's book on Pogonomyrmex. And so I got just really excited about Pogonomyrmex and started studying ants. And then I started into a lab. The lab I started in was a molecular lab. So these guys studied plants. One of them studied Gesneriaceae, the other some hybrid plants. And I got interested in hybridization and ended up in this lab running molecular markers and aldehydes. And that was pretty cool because I could help them out. But then I was the only guy studying ants. Everybody else studied raptors, and which look, they come back and say, look, I got some raptor scat out of it, you know. And they're like, well, I collected 50 colonies of today. So it turned out that it was a good subject to work on, and I became enamored with social insects and their inner workings, and so I went on to do a master's degree there, and then went to Arizona State, where I got in with Jennifer Fuel and John Harrison there. And that was really cool. But I kept studying the ants. So I was studying, different Pogonomyrmex and hybridization in Pogonomyrmex. And I was really excited about that. And so I finished that PhD and ended up in Diana Wheeler's lab and still studying the same thing. And this had not really run its course yet, but it was, for me, it was pretty much done. I was looking for a new project, and the turtle ants that she was studying, which are super cool, they're out here in Tucson area. And so I love to go out in the field. So I was out in the field, blah, blah. But the microbiome of this turtle ant became really interesting to me. And I started to run markers on the microbiome and try to figure out, what was going on with this particular species. And so that was what led me into this job. And then they announced this job here at Carl Hayden Bee Research Center. and I took the job studying the microbiome of ants, and it's been awesome. I've had really good lab members, really good postdocs, and really good support here at this lab to do this work. So it's been a lot of fun.
Cool. So you are in a very, very prestigious environment there. There is a lot of history there in that lab.
Oh.
And I think I only know about 2% of everything that lab have provide to the world. But I'm gonna keep my journey to learn as much as I can about that place. I visited once there. I think it was an American Honey Producers Association meeting, and then I had the opportunity to stop by and see the lab.
Right. Well, historically, that this has been considered a nutrition lab, and I've done some nutrition work. I mean, I was really hired to look at the microbiome of bee bread, which turned out to be a very vicious environment for microbes to try to survive in, because it's 50% honey. So it's kind of hard for anything to survive in 50% honey. But that was one of the first works that I did, was looking at that microbiome, and whether I was hopeful that it was going to be this nitrogen processing, microbiome. All of these things changed and the nutrition became better. And that's not what happens. It degrades, and then the fungus starts to eat it, and then the quality drops down. Yeah.
And that was the transition when you transitioned to honeybees from ants?
Yeah, that was, in Jennifer's lab, of course, it was during the Africanized invasion. And so I would help with them, with the honeybees, and which I considered an imposition at that time, because you were really, stepping on my toes, doing my ant work, So.
I know those USDA sometimes environments. The nutrition people cannot touch about pathogens, and the pathogens.
Oh, right.
Yeah, there is some fights on that sometimes. To me, as a spectator, it's a lot of fun to watch.
Oh, yeah. Often I feel less than prestigious. So.
Yeah. All right, Kirk. Let's jump into the article. Can we discuss about the last article you published? Can you give us a little overview about what it was, how it was done, and the results you guys got?
The one that was published in a peer-reviewed journal?
Yes, the peer-reviewed journal.
Sure. Well, first, I want to state that Randy was a huge part of this. So yes. Randy Oliver did all of the beekeeping, and we got together and sort of designed the experimental approach, which was, him, and with bees, as everyone who works with bees knows, you can get one result one time and another result another. But in this particular case, we used an enormous number of colonies. So I think we started with 78 colonies, and he did all this work. He triple blinded the study, which was awesome. So it was also during COVID. So we got, as you mentioned earlier, so we were getting samples. We weren't in the lab for about a year during COVID. So that's why it took so long to get the study out, to get all the molecular work done and through the process. But he set up, basically we had two different experiments. One of them is we fed the probiotic for many months, 5 or 6 months, to these bees, and then sampled them to see if they were any different than the control colonies. And it turns out that they were not different from the control colonies. And Randy, of course, the number one metric is, do you get more bees or do you get more honey, right? You get a bigger colony. And the answer was no. So he measured those things. And then he sent we did the fly-off assay, and he would pick bees that didn't fly off. And we got sent, tubes of those that we did molecular work on. I looked at the microbiome, and we looked at a number of disease markers, none of which differed. So we looked at nosema. We just looked at general fungal abundance. We looked at 4 different viruses: black queen cell virus, deformed wing virus A and B, and chronic bee paralysis virus. And none of those differed. Yeah, there goes the study right there. I recognize it.
Yeah, let me get to the figures here, and maybe you can explain using the figures here. That's gonna make it easier for the people at home to follow.
Oh, good.
We're talking.
Yeah, I'm just blathering about stuff.
No, that's fine. Just find a figure here. So in that figure, so tell me about what's going on here.
Yeah, this is associated with the antibiotic treatment. So in the second study, we took the same colonies and we subjected them to antibiotic treatment as they entered winter. And these are the results from that. So evenness on the top just tells you how structured the microbial community in the gut is, as the honeybee microbiota has incredible structure. And so you get very high evenness values, which you see in the controls across the board. You have very high evenness values. There was a dip. So down below, we ran qPCR on the 16S gene, which is really just a biomarker or a, what do you like to call that? Some kind of marker that tells you how much bacteria is in the gut.
Yeah.
And so those didn't differ across extreme. We had a little dip at 10 days, which may be due to, antibiotic moving around within the apiary. Who knows? But for the oxytetracycline and tylosin treatments, we had strong dips in the amount of bacteria that was in the gut, directly following treatment. That's the 10 days you see it down there on the x-axis. Both of those dropped off significantly, and then the evenness decreased massively. So what happens is you lose the bacteria that are not resistant to the antibiotics, and there are other bacteria that are part of the native system that move in that are resistant to antibiotics. So the gram-negative bacteria share these antibiotic resistance genes, and evolutionarily that's where they're suspected to have got it. But the bifidobacterium and the lactobacillus do not have these antibiotic resistance genes, so they get kind of wiped out. And then the other bacteria and who knows how efficient the microbiome is after it's replaced by these other kind of accessory or second string bacteria. So that's something that we're studying further. But yeah, basically what you see here is 33 days after treatment, the microbiome is still wiped out. You still have low evenness across the board at 33 days.
So what you're proving here is that antibiotic changed the microbiome, the quantities and the species on it. That's
yes. Yeah. And the evenness is the relative amounts of the species in comparison to one another.
Yeah. So the here you can see the big differences comparing from the evenness of the controls here for oxytetracycline and tylosin tartrate.
Right.
So big difference. So the antibiotics are affecting there.
We got a strong antibiotic effect.
Yeah. Okay, so let's go back here to
which is pretty well known, I think.
Yeah. Well, that's where we started any kind of, we need to make sure,
right,
especially in field trials, we need to make sure at least the things we know is still reproducing. Otherwise, you're lost in an ocean of variables.
Right. I don't think it's well known that it lasts that long.
Yeah, that's true.
That people don't measure that far out.
Yeah, so what are you watching here in this figure 2? So what's the main message here?
These are the controls. And what you see is the evenness that we were just talking about. And the evenness can be kind of determined by a gestalt in your mind. If you look at all the different colors and shapes, there's probably only one of these. You probably can't see my marker, but one of these that's all green in the pre-treatment.
You tell me that's it. One point.
Yeah, the one that's all green in the pre-treatment right there.
Pre-treatment. Here we go.
Got a big long green line down below.
And this one.
Yeah. So that's a dysbiotic microbiome. Right? Okay. The rest of them are very normal. And that's kind of what you would expect to see, is some variation along the line, not the dysbiotic. That's dominance as opposed to evenness, where you have the dominance of one or two species. In this case, generally there's like 13, maybe, they say there's 5 major groups. It depends on how you want to break them up, genera or species or strains, but you have this relative kind of abundance that you see in 7, 19, and 33 days.
So, and this is all the controls, right? There is no treatment here. You're just...
No treatment. Those are all of our control colonies.
This is one colony. This is another colony, another colony. And just one colony, you have a problematic colony here in the pre-treatment.
Yeah, that's an individual. So occasionally you get dysbiotic individuals. It's not that often. It's a very predictable, structured microbiome that's in the workers.
When you say individual, we're talking about is a pool of bees or...
These are individual bees.
Individual bees.
Yeah, each bar is an individual bee.
Okay. Cool. Okay, we established that. So there is with rare exceptions, things are not that changed between the bees inside the bee colony in this study.
That's right. Based on the tests that we use, these don't differ from each other.
Interesting. Okay. Let's move on to the next one. What are you watching here?
Yeah, this is a treatment with one of the antibiotics. Is this tylosin or this is oxytetracycline, I think.
Yeah.
Yeah, and right what you see happen is that the pre-treatment, we have that evenness that I was talking about, this even distribution of species, each color there being a different species that's listed in the box up top. And then after treatment, in the top panel, you can see there's a strong reduction in the number of bacteria.
Yeah.
And this starts to recover.
But then it's introduction on other things. Look at the passage. Look at
this is the alpha 2.1, also referred to as Commensalobacter, and this is a queen bacteria that is moving into the workers, right? And it is among the group of bacteria that tend to have antibiotic resistance genes. So Gilliamella has antibiotic resistance genes as well. So we see a big spike in Gilliamella apis that occurs after treatment, when usually it's at much lower abundance in the whole gut. And we see other bacteria popping up. So really you've kind of interrupted the structure of the microbiome. And it's this structure that we believe, or I believe, that really brings the functionality of the gut microbiome.
A question aside, since you mentioned that some of these bacteria that we find after the disruption very likely came from the queen. Do you know if the reverse happens sometimes? What happened with the queen? Do you know what happened with the queen's microbiota?
We've been working on the queen microbiota for quite a while. So it contains alpha 2.1 is the main bacteria. That's the yellow one here. This is the main bacteria in the queen's hindgut. But you can find it at low abundance in the workers, right? Yeah. So a lot of these things exist at low abundance. I mean, how does it get back to a new queen, right? It has to be transmitted with the swarm. So even the queen bacteria needs to go with the swarm, right? Correct. So here we have, that's the Bartonella. None of these are queen bacteria except the alpha 2.1. And a number of worker bacteria just don't occur in the queen, including Gilliamella. It doesn't like the Orbaceae. It doesn't like Gilliamella or Frishella apparently. So those two things it doesn't want in there. It'll allow Snodgrassella to hang out in its gut apparently. So we get Snodgrassella poking up in there. Yeah, I don't know if you want me to list all.
No. That's fine. I'm just trying to get the main message of the picture here and translate it in an easy way to my audience to understand the beekeeper. So yeah, like very easily to understand here, guys.
Yeah, visually, you don't have the order. So this is very ordered in the pre-treatment. And then you've lost all that order.
Yes.
And you have a bunch of colors which represent other bacteria that are now trying to dominate the system.
Yeah. And we have indications that this is problematic and could induce to health problems of honeybees.
Oh, absolutely. So yeah, this is tied to whatever you want to call it, lower fitness, lower health, lower growth.
Yeah. Cool.
Yeah, I think it's virtually the same for the tylosin. We have a little bit different pattern,
which is this one right here, right?
In the tylosin.
Yeah. So when we lost the amount, there is much less bacteria in the microbiome of those bees.
Right.
We're not talking about specific parts, right? Like this is a combination of the gut.
This is the entire gut. Yeah,
it's
the whole gut.
Okay, because we know there is some differences in quantities that could be biased from the rectum to the other parts of the midgut and things like that. But this is
the microbiome is highly compartmentalized.
Yeah.
But we put it together as a unit and they'll examine these. So we sequence the compartments separate from each other, and you get a very different looking picture. Of course.
Yeah, I just want to know what I'm looking at here.
This is the whole gut.
The whole gut. Okay.
Yeah.
Cool. So for you guys to understand again, with another antibiotic, the first thing that happened after 7 days, lost a lot of those bacteria. And then you can see by the different colors, which represents different species of bacteria here, there is an introduction and replacement of the original configuration of the microbiome of the honeybees with different guys. And what these different guys might be doing is very likely related with bad things with honeybee health. Is that a fair assessment?
Yeah, well, you've lost the structure, and the structure itself is the thing. These are native honeybee bacteria. Bartonella is a native bacteria, and it goes with the swarm. So does alpha 2.1. So this might not be the worst microbiome, but it's definitely more deficient than the normal microbiome.
Yeah, the structure, the way I like to talk with the people at home about the structure, what exactly that means, is try to bring this to terms that they understand in a daily basis. Like in a city, if you lose, we have a different kind of people, doing different kind of jobs for the city to work properly. Let's imagine you put some politics, decide to make a policy that's going to compromise the drivers. And then we don't have the drivers anymore. There is a replacement with other things, and the city starts to be chaotic and doesn't work the same way and could be bad, which is very likely is going to be bad in case you lose all the taxi drivers or things like that. And
exactly.
So that's
exactly it.
That's the way I try to explain to people why the change of structure could lead to problematic things.
Yeah, in this case, your taxi drivers have been replaced with people who really don't know the routes that they're supposed to go.
Yeah, with the AI taxi driver.
Yeah, or they may not be as friendly with the bus drivers.
Yeah,
as the other taxi drivers were.
Yeah,
they may run into the bus all the time.
Yeah. Gotcha. Cool. So interesting. So. And then you measure, you only mention in the text here that all other measurements didn't have anything related with the probiotics, right?
No.
Viruses, I don't see pictures or tables.
No, the paper, I didn't want to keep harping on the lack of, that's not what you do in a paper. You got a lack of effect. You say, well, we didn't have any effect, and you move on and you talk about things that you did have an effect, which was we had an effect with the antibiotics, which was
fair enough. Yeah.
The none of the disease markers showed any effect.
Or
the strongest data is probably Randy's. It's on his website, which is there's no change in colony size or colony weight.
And this was a different kind of, or Why it's not here? Is it a different publication from before?
He published it on his website. I mentioned that at the beginning of the discussion. It was not the data that was collected in this study. This was just a molecular study.
I see.
So I think that's at the beginning of the discussion, and then I cite his publication.
Okay.
So his, as far as I'm concerned, Randy's work is as good as many, many, many peer-reviewed publications that are out there. In fact, probably better because he's such an excellent beekeeper, right? And this is what's critical in bee research, as you know, is getting the front end of this experiment correct.
Yeah, I worked with Randy many years.
Yeah.
We have a big history. Randy is fantastic. I like Randy very much.
Yeah, I do too. He's quite a guy.
Yeah.
Great work.
Kirk, I want to ask you something. Let me explain my point of view on that. I did 2 field trials myself with some commercial available probiotics. As a private consultant for the beekeeping industry, sometimes commercial beekeepers hired me to do trials for them so they can keep this, they like to say, the secrets for themselves. So I performed once a trial for a commercial beekeeper, and I got exactly the results you're getting here. There was no difference at all with the bees. However, I was the scientist saying, look, after these calculations is everything we got, I don't see any statistically significant results here. The commercial guy said, you are out of your mind. I can clearly see there is a difference here. So, okay. So I let it go. You know, as a scientist, I did my job. I collect the numbers and I run the numbers, and I didn't see it too. I couldn't see. But because I respect beekeepers, he said, Umberto, you out of your mind. This is definitely have a difference here. So, okay, I don't want to fight. He paid for the trial. I give it everything to him, and we end up like that. That was my first experience with the probiotics out there. Said, okay, am I missing something here? Maybe the beekeeper can see something that I cannot, which is sometimes is true. You know, a person that is working in their own operation can know their bees a lot. So, okay, I let it go. Then years later, after when I was at the University of Florida doing research specifically for commercial beekeepers, one of these companies that commercialize probiotics, they paid a field trial using University of Florida to do a specific, very similar experiments. So I did it again, but in that time I was shocked that I got a result. The bees were heavier and wasn't statistically significant. I can tell you a little bit about the trial and the problems with the trial. Why I didn't publish is that it was in the middle of COVID. I lost all my students in the middle of the study. I have planned to capture all kinds of samples, to run all kinds of molecular analysis and all this jazz. But because, you know, with COVID, there is restrictions, the students cannot be together with each other, and then people go home. And that I was the only one doing the work, and I need to deal with 70 hives by myself. The only thing that I decide, okay, I need to just measure one thing. Okay, what's the main thing the beekeepers want to see? If the bees are heavier or not. So that's the one I focus on. And I got a statistically significant result. I was shocked. I said, what am I missing here? You know, I was sure I was not gonna get anything, and I got something. So I start to look at this whole probiotic things like, I will start to collect, okay, one more paper with evidence against, one more paper with evidence pro. And then when you look at the literature, you saw a lot of those things as you cited in your own publication here. I want to ask you, how do you deal with that especially with the field trials in honeybee research, that we have so many variables. How you feel confident with your field trials to pick one or another? How you do that? Because I don't know if sometimes I don't trust field trials as much because of the amount of variables that we cannot control, even doing the maximum things you can. I want to pick up your brain about how confident you are on the possibility that in radiologist environment, with the bugs they have there, with the treatments they have there, and blah, 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 we're not going to have a different situation that the probiotics could bring another result. Can you elaborate your thinking process regarding field trials and how you deal with that as a honeybee researcher?
Yeah, that's a tough question, especially for honeybee researchers.
Yeah.
There's a lot of stuff that I think the big story that I followed for a long time was this product called H13, and it is effective. It would inhibit American foul brood in a petri dish, and everyone was excited about that. And then they did a colony level experiment with it, and a robust one, and it didn't have any effect. And so I think that's more often the case, where you have these things that work in the lab, but somehow when you put them on the colony level, they don't seem to work anymore, and you can't really figure out why, because the colony is a very, very complex place, as you mentioned. It's overwhelmingly antimicrobial. I mean, this is a honey bee, right?
Yeah.
And the whole thing in there, and they go and they collect propolis, off of these populus trees and cottonwood trees, and they line the inside of that with it. And so I did a study with Marla Spivak about this, and there are bacteria. There seems that the native bacteria are not bothered by propolis, the native hive bacteria. So it's this Apolactobacillus conchii, Bombella, and Fructobacillus actually do better in propolis-rich environments. And all of it are all evolved to survive in honey. And if you don't have these, and they're part of what makes the environment nasty as well. It's like there's a little bit of real estate in there, but it's already owned by these specialists. And the same is true of the gut. There's this real estate, but any real estate that can be owned is already being owned by something that's highly co-evolved to be there and highly co-evolved to work with other species that are there. And that's why I think the big lab, there's a number of big honeybee labs now. Most of them spawned out of the Moran lab, which has just been an overwhelming boom for honeybee science. They've produced some incredible things and incredible researchers. So, you almost have to have a pre-adapted species. You have to have a native species if you want it to survive in this environment. You can't be introducing these non-native things. Not sure if I answered your question.
Yeah, I think the question is more like you gave an example. Like you have this I don't remember, some kind of chemical that in the lab conditions we find reproductive results. Reproducible results, and then you always go to the hives themselves, and we never able to get the same results, and very frustrating.
Yeah.
What I want to pick up your brain is, that if you do it again in a lab, you got the same result, but because the difficulties of field trials and how the scientific method is supposed to be, we have a tendency to look at the field trials as, okay, we didn't work in the field trials, therefore it doesn't work. How will you handle that? Because the scientific method is going to tell us that to be true, you need to reproduce. And we never do really field trials many, many, many times the same way, the same conditions. How you deal with this in your mind? as a honeybee researcher, because I never was able to be sure about things looking at in the field trials, or actually as a honeybee researcher, it's very hard for me to be sure about anything almost.
Oh boy, yeah, I understand that. Absolutely.
I wonder how you deal with that internally as a honeybee researcher.
Sure, I've had this conversation with everybody who researches honeybee colonies. It's just the way they are. There, it's very difficult to have something that you call a control. You're like, this is your control.
Yeah.
This giant thing that is moving like this and buzzing and has all these behavior and has all of these substances and constantly collecting and interacting with the environment all the time, right?
Yeah.
This is your control.
Yeah. Yeah.
Right? It's absurd. It's absurd,
exactly.
Right, to have a colony control. But it's very easy to do these inhibition assays or these things or PCR, and they get these very definite controls, and PCR will sometimes bother you, but generally you're going to get, similar results out of it. And that was the H13 I mentioned. They did inhibition assays in the lab, which are easy to repeat. Those H13 are the 13 bacteria that are already in the honeybee gut. They're already there.
Yeah.
And so they reintroduced those, and that didn't do any good. We did the same thing with the hive bacteria. We introduced Bombella, and this was some of this was published because it reduced nosema loads in one of our studies, which was cool. And but it didn't really result in any change in colony size, and so we did the colony level stuff. And again, we got no, it didn't seem to be doing anything. I think I mentioned that in the bee culture.
Yeah, well, we're going to get to that later. I want to talk about that later a little bit. But you mentioned before to me that you have in the past a field trial that you got some bees that were heavier using prebiotic. Prebiotic.
I think there was a bacillus in there.
Bacillus.
But we don't know what the effect was, or if it was the bacillus or the prebiotic. So this was with ApiCare. I shouldn't mention their product, but it's in a positive light. So I guess I could mention it, but
so you
can cut it out later.
You also had that same experience like me. Like you have a set of experiments that give you a suggestion from one side and a set of experiments that give you a suggestion for the other side. And that's why when I saw your letter, that I got curious that I'm missing something here, because in that letter, you were basically saying that, you correct me if I'm wrong, that the probiotics don't, that products are not useful at all. Is that a fair assessment? When I read, I felt there was some kind of emotional attachment to saying this is crap, basically. Is that the way you see it? Because when people ask me about probiotics, I feel like I don't know. Actually, I have this that work. I have these doesn't work. Honeybee research is complicated. I cannot say you anything.
Right.
That's it. But in that letter, I felt that you have a position on that you think is useless. Is that correct?
Well, part of the position there was the distinction between science and entrepreneurship.
Okay.
Right?
Yeah,
I think that Jay told me that it ended the last paragraph he might not have put into that. He might not have put into that article. And I said, and I went back and I read the last paragraph and was like, maybe he's right. Maybe I shouldn't have. That was a bridge too far.
Look, I don't mind. I have no, I think researchers need to go out like you more. To me, just write whatever you're thinking at the moment. That's fine. I like your style. Don't get me wrong. I just want to understand if I'm missing something here, because as a researcher, I'm not in the same category as you. You're doing this as a career, and I'm a consultant doing something, private studies in a field that I'm not an expert, bacteria. I'm a virologist from background, right. Not even comparable. But I'm always very cautious about trigger something that could minimize, the beekeeping industry itself is already, we need products, we need things to fulfill the things that are there. And beekeepers always ask, sometimes they're mad at me that I don't have a position. And I explain why I don't have a position, because I have two different things in my hand that I got different results. So I said, don't know. And when I saw your letter, I said, he knows. I want to know why. And I'm missing something because when I read the literature, I felt that, well, the literature is still like, we have evidence doing this, we have evidence doing that, respect to labs. But your letter gave me the impression that I am missing something. That's what I'm trying to do here. What am I missing for you to be sure that, not sure, but no, that's not fair. Let me put this back. Let me take this back. You are fair saying that entrepreneurship sometimes, actually a lot of times, messed up and only want to push for profit. We know them.
It just generates a bifurcation is what it does. I'm not saying that well, everybody's crooked who wants to make a
yes, I'm sure.
What it does is it generates a bifurcation. Like, I have desire now to make money.
Yeah.
And I have desire to do what's right. You have both desires. That's a bifurcation.
To do what's right. And I'm fair with that.
Sure. And making money is often, I think that's what's right. I need to make some money for my family and my kids. Yeah.
I also believe in individual accountability. If somebody decide, that's something that I do in my channel a lot. Look, I have a no position. Even when I have a position personally, sometimes I'm avoid to say it. I said, look, these are the facts that I know. You are responsible for your decisions at home.
Mm-hmm.
I truly believe in personal responsibility. I think we're lacking just with my kids. Like, that's your decision. Don't ask me to clean your mess. You clean your mess.
That's right.
People are looking for saviors and things like that. So I believe in personal accountability. So that being said, when the beekeeper make a decision to buy those products and he knows, or he didn't do a research to see that, we have research from this side. I also have research pointing to this side. So whatever decision he's going to make, he can't blame anybody other than themselves, right? And I think that was basically what I saw in the letter. Look, we have entrepreneurs, we have the science here, and I think that's where you're pointing in that letter. I love it when researchers go out and say whatever they want to say. It is I appreciate a lot, when researchers go and speak out like the way it is. Because sometimes we're right, sometimes we're wrong. And no, I just want to thank you to bring the point of the entrepreneurship, the dangers of that, pointed out the experiments that you have. No, that was an interesting way to see it.
I have no stake in it either way.
Yeah. But you don't...
If you want doing the research.
Yeah, but you don't believe there is, for example...
somebody who's not hopeful.
I got this question from this commercial guy, and I wanna bring it straight to you because my answer was, I don't know. So he asked me, Umberto, do you believe the probiotic might have a benefit in some sort of situation that that is not in those papers? My answer is, yeah. Yes, I guess, yes. That's what we're gonna say. Like in your example, the genetic of the bees, the rabies bees. I don't know if around pesticide too much. I don't know the conditions. Do I believe there is other conditions that the probiotics might have a beneficial effect? Yeah, I believe it, but I want to see it. I want to see data. I want to see results. And the only data and results that I have in my hands is 50-50, 2 trials that I did it myself. So what I tell you, I don't know. So what do you think? Do you think there is possibilities there? Or what areas of research would you like to see done to convince you that that maybe this area we might have something. There is any area that crossed your mind, or a situation combination of uh, of of um, variables that you think that maybe occasionally in this situation those probiotics might be beneficial here somehow.
The non-native probiotics, I would
Yeah.
say there's not much hope for non-native probiotics.
So that's your position. You don't have much hope on that.
That was pretty clear. I think they have. So I wrote about human probiotics, which by no means am I a master of that wheel of energy there, but there are conditions where human probiotics work, and they work well.
Yeah.
And a number of situations, but that's only about 5% of the things on the market.
Oh,
The rest of them survive through advertising, and they put out pseudoscience about what a microbiome is, and what a probiotic is, and here's why ours is better than this other one, and they're really not tested at all. So there's no regulation on these things, and so you can say really whatever you want to. You can make all these claims about what it does, but you don't have any information to back up what you're saying. So you're very right. Like, it needs to be a lot of information. There needs to be like an independent body that does this kind of work. So you can say, well, we had this independently tested, and that's, Randy's kind of an independent body, and so am I.
yeah.
I guess. And so there's certainly the data that we got from this experiment, which was we really looked with great detail to try to find any of these bacteria that were introduced as probiotics, to find some of these in the system. You can't even find them. And that's just the explanation for that is they do not survive the hive environment. That none of them are evolved to survive in the hive environment. Period. They don't they introduce them with sugar. So they're immediately, like any sugar, taken up into the crop of the worker bee, right? At that moment, they are dead. That's it for them, right? And it's amazing that we couldn't even really find the 16S sequence of any of these things. So none of them established anywhere in the gut.
Yeah, that was surprising to me, because right after to put, apparently everything disappeared, and you put it's a big quantity of the bacteria.
Well, we got 14 million reads. That's 14 million of these barcodes, each of them, from some type of bacteria that's in there. So it was a really good look to see if any of these things were in there. And D'Amico, this has been repeated. D'Amico did the same study.
Yeah.
And they could not find, I praised them because they sequenced the probiotic material itself, the stuff that you dump into the hive with the sugar, where the bacteria are supposed to be. And there are some bacteria in there. We grew them in the lab. There's a couple of orders of magnitude less than they say, and I think their yeast counts are still pretty high. But the D'Amico couldn't find any of these things in there either.
Yeah, those I follow those. This is something that I like, because when many different places are doing similar experiments and getting similar results, that's where I start to be more confident to go one direction than another. But what breaks my mind that I can't be sure about things is like situations like, okay, let's imagine, because most of the Randy's bees are pretty healthy.
Oh yeah, it's got good bees.
But that's the thing. I think do you think there is a situation that the bees are damaged by something, pesticide use or whatever pathogen, that already destroy the structure that you normally mention here, that those probiotics could be adding there and have a beneficial effect? I cannot say no to that possibility. That's why I never say to the beekeeper that I don't see it. I'm hopeless, like the way you are. You said you don't have no hope. I feel like when I talk with beekeepers, I cannot say I don't have hope, because maybe we need to simplify.
I just mean that I'm not hoping for a particular result.
Oh, okay.
I don't mean that I'm completely without hope or faith.
Oh, okay, got it. Okay.
Just not hoping for a particular thing to happen. right?
Oh, yeah, because it's something that crosses my mind all the time. Well, it's something that I cannot say, well, I don't know about that.
Yeah,
If there is damage,
you could interview some of these people from the Moran lab. So they're working, they're introducing, but this isn't the probiotic effect. This
yeah,
is something entirely different that you would understand well, given your background, probably better than me, and Jay would understand it too quite well. But they're introducing double-stranded RNA into the bacteria that they then introduce. So this is I forget what they call it, and you're just introducing a therapeutic. And
yeah,
in one case, I think they've knocked down varroa mites, so they get RNA interference in the varroa mite.
yeah. This is
The
the
vehicle to deliver that is one of the honeybee, native honeybee bacteria, the Snodgrassella bacteria, right?
I interviewed a gentleman, I forgot his name, that published in Science. I interviewed him in the past. That's a beautiful, beautiful piece of work. The problem with that is the acceptance of the people, because some people might consider that genetically modified, and then
Well, that is genetic.
it is genetically modified, and then is a whole level to bring this to the market is very hard when the people at home doesn't know too much about genetic modification. It's just like a tool. There is good ways to use it, and bad ways to use it, and yeah, it's another interview, completely right there.
It really is. It's an interesting approach. So, of course, they're going to try to use the microbiome as a vehicle to introduce all kinds of therapeutics and medicines. That's where the future is. That's what they're going to be doing.
Yeah.
No question. That's what already happened.
Yes. Yeah.
But that's not the microbiome itself.
No.
My position that the microbiome as it is pretty darn good. It is unbelievably amazing. There's really nothing like it in nature.
How surprised was you as a researcher when the people discovered that the gut microbiome was so simple with a few species? Because as a researcher myself, compared with other species, organisms that I look at the internet, at the PubMed, I was expecting the gut microbiome to be huge with hundreds of species, and then there is just a couple. How surprised was you at the time, and what's your thoughts on that?
I did not know at the time that what other microbiomes look like. So when I learned about that, and then Nancy Moran, I did a paper in 2012, and it was showing that the microbiomes of these two geographically distant groups were incredibly similar, right? Well, statistically indistinguishable, I guess would be a better way to put it. And then that really solidified, well, this is just the Apis mellifera microbiome. And it turns out to be, I mean, there's some variation geographically, but largely it's exactly the same species of, some places, and who knows if this is geography or just a particular experiment like we were talking about earlier. Where Bartonella is a lot more evident, and some of these other bacteria show up more often than they do. But generally, it's just the same thing. With humans, you can't find two people who have the same microbiome.
Yes, but the quantity is expected for comparing with humans, cows, but when I see the honeybees have a couple of species, I was shocked. But then after thinking about, well, maybe something related with the social environment, and there's a lot of honey there and a lot of, propolis, is supposed to be growing only very tough, specific bacteria that co-evolve with them for thousands and thousands of years. It's not an environment that easy for microbes to grow.
No, it's evolved with this incredibly, so stole things apparently from the foraging environment and the floral environment.
Yeah.
And those things became more and more able to live just in the honeybee, and then moved with the swarm. So the swarm is, I think, the essence of why it is so particular and so predictable, is that swarm has had contact with every swarm back through the ages. That's how it's transmitted, swarm to swarm to swarm.
Yeah.
And it's nothing but, that swarm you're holding on to stretches back through the eons
Wow.
in terms of contact, right?
Yeah.
Where you have thousands of functional microbiomes in every worker in that swarm.
Wow. Yeah, honeybees keep amazing me every time. It's
I've always loved social insects, but studying the microbiome of this one of the most unique social insects has just been, it's been so much fun. I've really enjoyed it.
Is this a feature of only honeybees or all social insects? To have a very limited amount, species-wise, number of species in the gut microbiome.
Yeah, you don't see anything like it. So these things that have to overwinter, there's a number of species that are individual. They get put in a nest. The bees get put into solitary bees get put into the nest, and whatever ends up in their larval development and so on. And there's nothing that has this budding, if you have this budding structure, that's where I think you have the potential for this type of evolution to occur with the microbiome and the host. The others where you have this very distinct, I laid an egg and something got smeared on that egg, and how well does that thing move into the adult generation, right? What we have here is we have direct contact with adult generation to adult generation to adult generation that never stops.
Yep.
Right. And that's, I think, what's allowed this evolution of such a particular and beautiful microbiome.
Yeah.
And no, I don't think it needs any help.
When they're intact, if they're damaged, maybe we need.
Yeah, there may be combinations of various strains that work better than others, a little bit better, but all those strains are still within that group. And how are you going to reintroduce them into that group? Well, they're already there. And the same thing with, the Stagracilla, you're going to put it in there. It's going to get replaced by a more efficient Stagracilla. You're going to have to make it very efficient at what it does for its job as part of the microbiome and the host interaction, right? And then on top of it, put this energetic constraint of you have to pump out double-stranded RNA all the time, right?
Yep.
Is that going to be okay with that system? Is that taxi driver going to be all right?
That will be something very interesting to see.
Yeah, I don't know. And I think, I don't know. I think everything's going to be done, of course, and they're going to figure out a way to do it all.
Yeah. Kirk, before I wrap this up, I want you just to know a little bit, what other projects are you doing right now with the Bee Lab there that you want people to know?
Oh, we've had a huge project. It's an AI project diagnosing brood disease. So we've got all of these incredible images and molecular signatures of brood disease, and we're trying to turn this into an application where you could just take a picture of your brood disease. And we'll be working with Mohammed Al-Baraki. I've just been chatting with him a bit. If you know Mohammed, he's really cool. He's got a lot of great ideas. So we're going to try to make this app the beekeepers can use to just.
You take a picture of the frame and then you diagnose the brood disease,
and you can diagnose disease with just a picture.
Interesting.
Well, it's a bit difficult because there's a lot of this. We started with the most difficult kind of determination, which are these blobs of larvae, right?
Yeah.
And so you could get a blob-looking larva for EFB, and you can get a blob-looking larva from virus, but we've got it trained pretty well to be able to distinguish the virus from the EFB, and it does way better than, I think, the gestalt of a person. But who knows? There's lots of skeptics about AI too. It's all about how you use it and what you use it for, right?
It's a new tool, like with some tools in energy, we can create good things or atomic bombs. It's up to us to decide how to use the tools.
Yeah, Dwan Copeland, so he's my postdoc. He's been really championing this work. He's been working super hard, moving this down the field. He's impressive.
So just tell him I'm gonna send him an email to talk about this project.
Okay.
in the future. Yeah, that would be great.
It's really cool. He would love that.
All right, Kirk, do you have any message for beekeepers out there, about the work you're doing or the mission of the lab? You have the open floor for you.
Yeah, I would say relative to what we've been talking about, if something works for you, do it. I've heard stories like, well, that can't be true because I've been using this and it works well for me. Well, if that's true, keep doing it if it's working. And the second thing you should do is set up a situation where you can do your own research, right? Where you can discover your own truths. And if you need to work somebody, Randy got, I think he donated, not donated, he gets donations from beekeepers. He gave us, so I think $8,000 to help with this work. And it was a good amount to help get some of this work done. It doesn't come cheaply, right?
Yeah.
I bet you don't come cheaply either, Umberto.
I would, I need to survive. I need to say that.
Right.
So it's hard work. The bees, yes, and it's a lot of work. Yeah. But the commercial guys that hire me, they have the means and the people that could help me. So for them, doing it for themselves, just paying for my time, it's not a big deal for me to organize and make the things scientifically, accurate. But the hard work, maybe it's just me with the workers, just observing and they keep the truth for their operation. That's something that I think is important. The commercial guys out there, there is so many situations when I do experiments specifically locally. That's very likely it's going to be something that might be repeatable over time in your operations. You never know when you have one species of plant in your neighbor that's going to mess up your own operation.
Right.
So, yeah. So if you want to hire me, I'm here.
Awesome. Well, it's been great to talk to you, man. I thank you very much for this opportunity.
My pleasure. And I'm looking forward to meet you one of these days and have more conversations about the rest of the projects you're doing. And thank you to put all this effort out there to show us the things that, that nobody want to see it sometimes.
You're welcome, Mike. You think Randy's the one that really started this.
I'll bring Randy here to discuss this too. Yeah. I'm gonna invite him.
Yeah. I will take full credit though. You can give it to Randy.
Okay. Kirk, thank you very much for your time, and we from Inside the Hive TV really appreciate. Thank you.
All right, thank you.
All right, and for you guys at home, thanks for watching. See you guys in the next video. Inside the Hive.tv, the show about peace. Bye-bye.
Thank you.